Okay, I'm starting the class. So, as I discussed that uh, we will be talking about spermatogenesis. Are you people able to see this?
Yes, sir.
Yes, sir.
So, spermatogenesis, it is the process by which haploid spermatozoa develop from germ cells in the seminiferous tubules of the testes. This process starts with the mitotic division of the stem cells located close to the basement membrane of the tubules. Meiosis is a step during spermatogenesis and oogenesis. Spermatogenesis produces four haploid sperm cells, while oogenesis produces one mature ovum. So you have haploid, that is having n number of chromosomes, which develops from the sperm mother cells or germ cells, germinal cells, from where germination takes place. At least it happens in the seminiferous tubules of the testes. This process starts with the mitotic division of the stem cells. The stem cells are the precursor mother cells. The stem cells are the precursor mother cells. From which matures and forms the sperm mother cells, and from where spermatogenesis takes place, and you have a spermatozoa or sperm being formed. And there is a process of meiosis or reductional division which takes place here. In spermatogenesis, you have four haploid sperm cells present, while in oogenesis, one mature ovum is present. But the other three, which are supposed to be formed, they go out as polar bodies. They don't form eggs. So two haploid spermatids or haploid cells are generated by each secondary spermatocyte. So you have a primary spermatocyte which makes two secondary spermatocytes. Each of the secondary spermatocyte makes two spermatids. And through the process of spermiogenesis, the spermatids, they resemble the spermatozoa or the sperm with flagella. There are changes which take place in the spermatid. The spermatid looks like a normal cell. The seminiferous tubules in which the sperm cells are produced constitute about 90% of the testicular region. The immature cells, they are also known as spermatogenia, are derived from cells called stem cells in the outer wall of the seminiferous tubules. These stem cells are sperm mother cells. So on the these small tubes which are there, they are on the walls. So the mature sperm comes towards the center. The process of producing mature sperm is called spermatogenesis. Sperm are produced in the seminiferous tubules of the testes and become mature in the epididymis. The entire process takes about 9 to 10 weeks for sperm to uh, form. Now, the spermatogonium or sperm mother cells, mitosis takes place and it forms two spermatogonia. There are different A, B type of spermatogonia. And this spermatogonia, it forms primary spermatocyte. They form primary spermatocyte. This is also 2N, this is also 2N, 2N. Then here the primary after meiotic cell division 1, it forms N. And then meiotic division 2 takes place and spermatids are formed. Then through the process of differentiation, spermiogenesis, these spermatids, they come and they become spermatozoa, and then they're released and the residual bodies remain behind. So this is the cross section through the seminiferous tubule. Tube, the cross section. So this is the basal lamina surrounding the seminiferous tubules. In between, you have the Leydig cells. The spermatogonium is absolutely in this region. And as you go in the lumen towards the center, you'll have the spermatozoa over here. So basal lamina, you are taking a section and doing the spermatogonium. Then here it is the spermatocyte primary. Then the secondary spermatocyte, which forms the spermatid. And then that differentiates into the spermatozoa and that is released inside. And the residual body is remaining here. You have lots of residual bodies staying here. So this is how the spermatogenesis actually takes place, where it takes place. Clear so far? Yes, no?
Yes, sir.
Okay.
Yes, sir.
Okay. So you are taking a cross section, you are looking at a further magnification. Here you have the basement membrane, the sustentacular cells, which these cells, they sustain this spermatogonia, they give enough nutrition to them. There is a blood testis barrier. These spermatogonia here are known as type A spermatogonia from the basis of their color. Then here it forms the type B spermatogonia. Then these are the primary spermatocytes, which are still 2N. The secondary spermatocytes become NN, and then second meiotic division takes place. The spermatids are formed, and from there you have the mature sperms formed. So let us look at spermatogenesis, the stages. So one daughter cells remains at the outer edge of the seminiferous tubules to maintain the germ cell line. And so from the sperm mother cell, one becomes, it remains, so that in the next cycle, again, more spermatogonia is formed. So one daughter cell moves towards the lumen to produce spermatozoa. So it has got 46 number of chromosomes, and that is 2N, diploid number. So, and one is making spermatozoa. So this one makes 2, still 46 and diploid. The primary spermatocyte takes place. First meiotic division takes place. This 2N becomes N. Second meiotic division, which is same as mitosis, you have so many 23. They are haploid number of spermatids being formed. And finally, you have the matured spermatozoa being formed. Formed. So these are the steps you can see. Spermatogonia, does meiosis takes place, and then gradually the spermatozoa is formed. The steps of spermatogenesis. Now we are going to spermiogenesis. So first, what happens appear from the spermatids to the spermatozoan. We are seeing you have the Golgi complex over here, and acrosomal vesicle is formed, and a bit of a basal body from where a flagellum is there. So there is appearance of acrosomal vesicle and flagellum in the spermatid. Now the acrosome is now covering the nucleus. It's growing. The Golgi is getting converted into the acrosome, and the flagella is increasing in size. Gradually, the excess cytoplasm is removed, and the acrosome is formed. Finally, lots of mitochondria known as axons are formed over here. Here, this is the mid place. This is the acrosome formed from the Golgi body, and the mature sperm is formed. This is the process of spermiogenesis. The first phase is the Golgi phase. Glycoprotein-rich granule acrosomal vesicle is formed. Mitochondria are displaced peripherally. Side dike chole gel. Centrioles migrate towards opposite poles. Distal centriole begins to form axoneme. Axoneme হচ্ছে এই মাঝখানের এই জিনিসটাকে axoneme বলছে. The next is the cap phase, where the cap is being formed. And then the acrosomal phase, where the acrosome is formed. Excess cytoplasm is shed out. So this is the spermatid phase. The same thing, matured sperm is formed. So if you look at the hormonal control of spermatogenesis, so formation of primary spermatocytes and entry into early prophase ones begins during embryonic development. And then spermatogenesis is arrested till puberty in prophase 1. Testosterone and its metabolites are required for the completion of meiosis and spermatid maturation. You need the hormone testosterone. FSH is not essential for spermatogenesis, but it enhances T-stimulated spermatogenesis. At puberty, it acts with T cells to stimulate proliferation of cytoplasm. You need FSH for other functions, which make sperm formation. Directly for spermatogenesis, it is not needed. You don't need to study this part in developmental biology. Okay. Do you people have questions? বুঝতে পেরেছো? হ্যাঁ না? আচ্ছা,
Yes, sir.
ঠিক আছে। তাহলে বাই বাই। টাটা, আবার নেক্সট ডে ক্লাসেতে আমি বলে দেবো।
স্যার কবে নেবেন আবার?
আবার বুধবার দিন তো ক্লাস, বুধবার নেব।
ওকে স্যার।
Thank you, sir.
ওয়েলকাম।
থ্যাংক ইউ স্যার।
ওয়েলকাম।